All right. To continue on in our notes about cell division, so we talked about interphase, mitosis, cytokinesis, and now we're going to get more in depth with the stages of mitosis. First one being prophase. The chromatin coils into chromosomes. Identical strands called chromatids are held together by a centromere, and centrioles will direct assembly of the mitotic spindle. Puts those spindle fibers out, connects to the centromere, lining them up to the middle of the cell so that they can be split evenly. The nuclear envelope and nucleoli have broken down in this stage. Next comes metaphase. Chromosomes are aligned in the center of the cell on the metaphase plate. Again, by the spindle fibers attaching to it and moving them so that they're lined up in the middle. And so it appears like it is a straight line of chromosomes found in the very center of the cell. Next comes anaphase. This is where the centromeres are splitting apart and the chromatids move slowly towards the opposite ends of the cell. And anaphase is over when the chromosomes stop moving. Remember anaphase, and the chromosomes split apart, anaphase apart. Once anaphase is complete, telophase begins, and it is basically the reverse of prophase because the chromosomes begin to uncoil to transition back into chromatin, like the spaghetti-looking DNA. Spindles will break down and disappear until they are needed again. Nuclear envelope will reform around the chromatin, and the nucleoli will appear in each of the new daughter cells, daughter nuclei. And occurring basically simultaneous to telophase is Cytokinesis, the division of the cytoplasm, begins during the late anaphase and completes during telophase. Creates a cleavage furrow that pinches the cell basically into two separate parts, and then creates the two new daughter cells then. Usually most of the time, mitosis and cytokinesis will both occur together. So if mitosis is happening, cytokinesis is going to also happen. However, sometimes that is not the case. Then you get binucleate or multinucleate cells. And this can happen and is normal for cells that make up the liver and then also our skeletal muscles. So this is just the picture that is in your book on page 84, showing each of the stages of mitosis. These next slides show each of the individual ones. So prophase, see the chromatin there, you have the centrioles that are starting to form, and then the nucleus is going to start to break down. Spindle apparatus are migrating to opposite ends of the cell. They're attaching to the chromosomes to line them up in the middle of the cell during metaphase. So, and that it just ensures that they are being split evenly when they line up in the middle, so that the pull from each side is equal. Anaphase, they're being split apart from each other. Here we have telophase and cytokinesis happening. So we get this pinching. It's kind of like putting like a belt or something around like a balloon, and it just keeps getting pulled tighter and tighter and tighter. Eventually it pinches off into two new cells then. And then you have the nuclear envelope reforming, and your chromosomes are going to start to relax into that chromatin form again, where it's entering back into the interphase stage, where it cell spends the majority of its time in doing its normal activities. Okay, I know that was pretty quick, but that is a review of stuff we did in biology, as is this protein synthesis that we're going to talk about. So probably go through this relatively quickly as well. So DNA is the blueprint for making proteins. Proteins are basically what is responsible for our body functioning as a whole. If we didn't make the proteins, then we would not be able to do basically anything. Have to have those proteins to serve different functions, different jobs. So, on our DNA, there are several genes. There is a lot of our DNA that is completely useless. It's just kind of a placeholder there to keep genes separate from each other. So it's not just continuous run-on of those genes. So, gene is a specific segment of DNA that carries a blueprint for building one protein or a polypeptide chain. And proteins have a lot of different functions. You have fibrous or structural proteins that are the building materials for cells. Or you have globular proteins, which are the functional ones that can act as enzymes or biological catalysts for all of the different chemical reactions that must take place in your bodies in order for you to function properly. So DNA information is coded into a sequence of bases. This is a specific sequence. Like those arrangements of your G's, your C's, your A's, your T's, they are put into a specific sequence to create a specific protein for a job. And then we have a sequence of 3 bases, a triplet. Codes for an amino acid. Anybody remember what those are called? Those triplets of DNA bases? Called a codon. So a codon codes for a specific amino acid. So an example here, DNA sequence of AAA specifies the amino acid phenylalanine. And you guys remember using the codon chart. It was the wheel. Remember the wheel that had the letter in the middle, and then you worked your way out until you found the amino acid that it coded for. So, for the role of DNA, what it's actually doing, most ribosomes are which are the manufacturing sites of proteins, are going to be located in the cytoplasm of a cell. Most of them, not all of them, because some of them are attached to the endoplasmic reticulum. The DNA never leaves the nucleus in interphase cells, so while it is carrying out these functions, these normal functions of producing proteins, the DNA is always going to be in the nucleus. Only time it ever comes out of the nucleus is during that mitosis process where the nuclear envelope disappears. But very short period of time that happens. DNA requires a decoder and a messenger to carry instructions to build proteins to the ribosomes. And both the decoder and the messenger functions are carried out by RNA, which is ribonucleic acid. So we have, remember there are 3 types of RNA, which we're going to talk about again: mRNA, tRNA, and rRNA. And those basically get the instructions from the DNA that allow for the synthesis of the proteins. So DNA tells them what to do. RNA actually does the job of making the proteins. Slight differences between our DNA and our RNA. If you remember that RNA is single-stranded rather than being a double-stranded helix like DNA. And RNA contains a ribose sugar instead of a deoxyribose sugar. And RNA contains uracil base instead of the thiamine base. Are three types of RNA: transfer RNA, ribosomal RNA, messenger RNA. tRNA transfers the appropriate amino acid to the ribosome for building the protein. Page 87 in your books does show a very nice visual of the protein synthesis process. And it has tRNA molecules in there that bring in the anticodon, so opposite of the triplet of our RNA then, with a specific amino acid on it, and brings it to the ribosome, where then it helps with building that chain of amino acids that will become our protein. Ribosomal RNA, rRNA, helps form the ribosomes where proteins are built. And then our messenger RNA carries the instructions from the nucleus to the ribosome. So it gets the message directly from the DNA. It is a complementary strand of RNA to that DNA strand. And it takes that message to the ribosome, where the tRNA brings in the amino acid. The two processes of protein synthesis are transcription and translation. So transcription occurs first. This occurs in the nucleus of the cell. Transfers the information from DNA's base sequence to the complementary base sequence of mRNA. DNA is a template for transcription. mRNA is going to be the product of that. And it is a complementary strand. Each DNA triplet corresponds to an mRNA codon. And so if our DNA sequence is AAT, CGT, TCG, the mRNA, complementary mRNA strand or codons then would be UUA, GCAAGC. And I'm sure you guys remember doing that, right? So that illustration on page 87 shows that whole process, transcription, again, taking place in the nucleus. So you have your DNA at the site of origin. The DNA is basically going to unzip at that spot where the gene starts, which allows for this sequence of mRNA to be made. Okay, once the mRNA is made, it can leave the nucleus. That DNA zips right back up. Translation has a few more steps to it than what transcription does. So in translation, the base sequence of nucleic acid is translated to an amino acid sequence, and amino acids are the building blocks of proteins. And this will occur in the cytoplasm and involves those three major varieties of RNA. So tRNA, mRNA, rRNA. Are you kidding me?